Alright guys, we'll be having an overview on gram stain and talking about gram stain. We spoke about we spoke a little about gram stain when we were talking about the types of bacteria, okay? And we said that bacteria are also classified based on what gram staining, okay? That's where we're talking about gram positive staining bacteria and gram negative staining bacteria and all have you, right? So in this lecture, we'll be talking about this whole concept of gram staining, right? Its uses and its applications, all right? And how it is done, all right? So this is the gram staining technique lecture, all right? So you could say this is like an overview of gram staining, all right? I feel like let's go into the lecture before we come back. So basically, guys, gram staining is a fundamental technique in microbiology and helps to differentiate bacterial species based on their cell wall properties, all right? So here now you get to see if the bacteria is thick cell wall, what is it called? If the bacteria is thin cell wall, what is it called? Do you understand? What is the principle of gram staining? We said that Danish bacteriologist Hans Christian Gram introduced this technique in 1884. All right, so it classifies bacteria into two groups. That's the gram-positive bacteria and the gram-negative bacteria. Okay, so this stain process involves primary stain. That's the crystal violet. We have the mordant. That's iodine, and we have the decolorizer, usually with an alcohol or acetone, okay, and also we have a counter stain that's a safranin, okay. So if a bacteria is a gram negative, it should be decolorized, okay. I remember that from my experiment days. So the key difference lies in the structure of their cells. Gram positive bacteria have a thick layer of protein sugar complexes called the peptidoglycans, and they have a low lipid content. Now, the thick cell wall retains the crystal violet stain appearing blue or purple, right? So it is not decolorized, right? So it's a gram positive bacteria. Then for the gram negative bacteria, on the other hand, they have a thin peptidoglycan layer and they have a thick outer lipid layer. All right, so now the crystal violet iodine complex gets washed off during decolorization and they take up the safranin stain appearing red. Okay, these are gram negative bacteria. Okay, so you can see this is a gram positive, this is a gram negative. So from the fixation to application of crystalline violet, that's a primary stain, to the iodine treatment, to the decolorization, you could see that the gram-positive bacteria, they will retain the color, they will not be decolorized, right? But for the gram-negative bacteria, they will be decolorized and they will take up the color of the decolorizer, that's the red color, right? So what is the procedure like? We said that prepare a clean slide with a bacterial smear. Apply crystal violet for 30 seconds to 1 minute, then rinse it. All right. Add grams iodine for 1 minute and wash. Decolorize with 90% alcohol or acetone. All right. Now apply safranin for 1 minute and wash. Air dry, blot dry, and observe under a microscope. Okay. So what are the examples of gram-positive bacteria? Examples of gram-positive bacteria, we have the Actinomyces, we have the Bacillus, we have the Clostridium, we have the Corynebacterium, we have the Staphylococcus, we have the Streptococcus, and more. Okay, so guys, this is basically the gram staining procedure, talking about the principles, uh, also talking about the procedures. All right, so guys, that's it about what you guys need to know about the gram staining technique. But I think I'll still be having a video on the procedures, right? Where I'll be showing the experimental basis of gram staining. All right. So guys, that's it about the basics you guys need to know about gram staining technique. Basically, it's used to differentiate bacteria. So know if it is a gram positive bacteria or a gram negative bacteria. Okay. So that's it, and bye for now.